Hi, myself Gayatri from the Department of Zoology, BSc CAS, Coimbatore. In this video, we are going to see about the bacterial insecticide Bacillus thuringiensis. Look at the classification. It comes under the domain Bacteria, phylum Bacillota, class Bacilli, order Bacillales, family Bacilliaceae, genus Bacillus, and finally it comes under the species Bacillus thuringiensis. Let us see the structure of the cryproteins. Cryproteins usually have the three domains, as you can see in the picture, domain 1, 2 and 3. Domain 1 contains the 7-alpha helix which helps in the membrane insertion. Domain 2 consists of the beta prism of 3 antiparallel beta sheets which helps in the receptor recognition. Domain 3 consists of the beta sandwich of antiparallel beta sheets. Okay, let's start with the discovery first. In 1902, Bacillus thuringiensis was first discovered in the silkworms. It was discovered by the Japanese sericultural engineer named Ishiwatari Tsujitani. He named it as the Bacillus soto because the Japanese word soto means collapse. Here it refers to the bacillary paralysis. In 1996, another class of the insecticidal proteins in BT was discovered. It is a vegetative insecticidal protein which is present in the Bacillus thuringiensis. In 1911, German microbiologist Ernst Berliner rediscovered it when he isolated it as the cause of a disease called scallopshire in flower-mouthed caterpillars in Thuringia. In 1976, Robert A. Zakarian reported the presence of a plasmid in a strain of Bacillus thuringiensis and suggested the plasmid's involvement in endospore and crystal formation. We see about these toxins in the upcoming slides. As of now, you remember in 1976, Zakarian reported the presence of a plasmid in a strain. Then in 2000, a novel subgroup of cryprotein, which we are going to see in the upcoming slide, what is meant by cryprotein. So, designated the parasporin, which was discovered from the non-insecticidal Bacillus thuringiensis isolates. Then, here our topic comes here. Bacillus thuringiensis. Bacillus thuringiensis, which is also known as the BT, is a gram-positive bacteria, soil-dwelling, and it is most commonly used as the microbial pesticide all over the world. Bacillus thuringiensis also occurs naturally in the gut of the caterpillars of various moths and butterflies, as well as on the leaf surfaces, aquatic environments, animal feces, insect-rich environments, and flour mills and grain storage facilities. What are the toxins produced by the Bacillus thuringiensis? Here are the four main toxins produced by Bacillus thuringiensis. First one is alpha endotoxin, second one is beta exotoxin, third one is delta endotoxin, fourth one is loose factor. During the sporulation, many BT strains produce the crystal proteins, which are the proteinaceous inclusions called as the delta endotoxins, which comes under the category 3, which is mentioned above, that have the insecticidal action. This has led to the use of the insecticides and more recently to genetically modified crops. We know the BT cotton. Now BT corn is also available. Monsanto responded by introducing a second generation cotton with multiple BT proteins which are rapidly adopted. Bollworm resistance to first generation BT cotton was also identified in Australia, China, Spain and the United States. Additionally, the resistance to BT was documented in field population of diamondback moth in Hawaii, the continental US and Asia. Studies in the cabbage lupus have suggested that a mutation in the membrane transporter ABCC2 and can confer the resistance to BT cry1AC. What are cryproteins? The proteins that Bacillus thuringiensis is most known for are encoded by the cry genes. So cryproteins are encoded by the cry genes and these are located in a plasmid. As a toxic mechanism, these cryproteins bind to the specific receptors on the membrane of the midgut, especially to the epithelial cells of the targeted pest. Example, the targeted pest of the cotton is bollworm, which result in the rupture of the midgut epithelial cells. Other organisms like humans and non-targeted insects and animals lack these appropriate receptors in the gut. So they are not affected by the cryprotein and therefore they are not affected by the BT. Cryproteins and their mechanism of action. So Bacillus thuringiensis serves as an important reservoir of cryotoxins for the production of biological insecticides and insect-resistant genetically modified crops. When insects ingest the toxin crystals, their alkaline digestive tracts denature the insoluble crystals, making them soluble and thus amenable to being cut with proteases found in the insect gut, which liberate the toxin from the crystal. The cryotoxin is then inserted into the insect gut cell membrane, paralyzing the digestive tract and forming a pore. The insect stops eating and starves to death. Live Bt bacteria may also colonize the insect, which can contribute to death. Upon sporulation, the Bacillus thuringiensis form the crystals of two types. One is the delta endotoxin, which I mentioned in the previous slides, and the other one is CYT proteins. The Bacillus thuringiensis, or the small RNA, which is called as the BTSR1, can silence the Cry5BA toxin expression when outside the host by binding to the RBS site of the Cry5BA toxin transcript to avoid the nematode behavioral defenses. The silencing results in an increase of the bacterial ingestion by the elegans. The expression of the BTSR1 is then reduced after ingestion, resulting in the Cry5BA toxin production and host death. Some isolates of Bacillus thuringiensis produce a class of insecticidal small molecules called as the beta exotoxin, the common name for which is Thuringiensin. A consensus document produced by the OECD says beta exotoxins are known to be toxic to humans and almost all other forms of life, and its presence is prohibited in Bacillus thuringiensis microbial products. Thuringiensis are nucleoside analogues. They inhibit the RNA polymerase activity, a process common to all forms of life in rats and bacteria alike. So what are the challenges involved in this Bt protein? So multiple insects have developed a resistance to Bacillus thuringiensis. As of study in November 2009, Monsanto scientists found the pink bollworm had become resistant to the first generation Bt cotton in parts of Gujarat, which is in India. That generation expresses one Bt gene, Cry1AC. This was the first instance of Bt resistance confirmed by Monsanto anywhere in the world. So here comes the application of the Bt proteins, Cry-proteins. So Bt cotton is the first application, we are all familiar with that. Bt cotton is nothing but it is a genetically modified variety of cotton that produces an insecticide against the insects like bollworms. It is effective against the plant bugs, stick bugs and aphids. It came for the commercial use in the year 1995. Bt cotton in India. India is the largest producer and consumer country after the China when it comes to the cotton. In 2002, a joint venture between the Monsanto and NACO introduced Bt cotton to India. India has the largest hectareage of cotton and accounts for approximately 1/3 of the total cotton that is planted all over the world. So here is the illustration of a healthy and damaged cotton flower. On your left side you can see the damaged cotton flower, and on your right side you can see the healthy flower. And this healthy cotton flower is after the application of the Bt formulation. So before the application we can see the damaged cotton flower, which leads to the decrease in the production of the cotton. The other applications of Bt is here you can see the peanut leaves. So on the top dish the peanut leaves are destroyed by the cornstalk borer larva, and on the bottom dish you can see the healthy leaves, and this is due to the application of the Bt proteins. The other example is Aedes mosquito. So Aedes mosquito, we all know it spreads the yellow fever. So this mosquito can be controlled by the application of the Bt formulations. So the crystal of toxin, so we call it as endotoxin or delta endotoxin. So delta endotoxins are used in the destruction of this Aedes mosquito, so the yellow fever can be controlled. So these are the applications of the Bt proteins. So let's have a quick summary. We saw about the Bacillus thuringiensis, its classification, and what are the toxins produced by Bacillus thuringiensis. We saw about the Bt genes, Cry proteins, and what are the challenges we are facing here in India and all over the world while applying this BT formulation. So we saw about the challenges, then we saw about the applications, that is BT cotton and peanuts, and how the BT formulation is used in the Aedes mosquito to reduce the yellow fever. So this is all we saw in this video. So thank you all and have a great day.